Hello, friends. Welcome to another session of Biochemistry by Dr. Nilesh Chandra. And in this one, we are doing the quick recall for questions asked in November 2021 INI-CET from the Biochemistry section. As we are doing in all the papers in this series, we are only going to be looking at the questions, their options, and the correct answer. We are not going to do the discussion for this. We have already shortlisted the 60 main topics and 12 additional topics which are being asked in INI-CET over the last 10 papers. We'll discuss each of the topics separately once we've done the recalls for these papers, at least till 2020. Okay, so let's quickly do the questions, their options, and the correct answer that you should be knowing. Starting with first question, which of the following consumes the highest energy during assimilation? So here your options are proteins, carbohydrates, MUFA, and PUFA. What you have to remember that the highest energy during assimilation is consumed by the proteins, which is approximately 30%. We'll have a separate video discussing the thermic energy of food, and we will talk about all these aspects in great detail. Calorific value, thermic energy of food, all of that will be discussed in that video. Moving to next question, which of the following is not amphipathic? Your options are glycolipids, phospholipids, sphingolipids, and the triacyl glycerol. Okay, please note the glycolipids, phospholipid, and the sphingolipid. These are complex lipids. They are amphipathic. Triacyl glycerol is not hydrophilic. It is hydrophobic. It is not hydrophilic. Therefore, it is not amphipathic. The correct answer here is triacyl glycerol. Moving to next question, Wobble hypothesis explains which of the following concepts of the genetic code? Your options are degeneracy, unambiguous, non-punctuate, and universality. Please note the Wobble hypothesis is responsible for what is known as a degeneracy, means multiple codons code for the same amino acid. This is possible only because of the Wobble hypothesis. The correct answer is degeneracy. Next question, a patient with thiamine deficiency develops acidosis. Which of the following enzymes was affected? Your options are pyruvate carboxylase, pyruvate dehydrogenase, the PEPCK, and the lactate dehydrogenase. Please note the correct answer here is the pyruvate dehydrogenase. This is because thiamine is required as one of the coenzymes for pyruvate. We will discuss this when we're talking about the pyruvate dehydrogenase complex, that 5 coenzymes are required, one of them being the thymine pyrophosphate. So in that video, we'll talk about all the cofactors, the 3 enzymes which are required in the pyruvate dehydrogenase complex. Question, which of the following tests will be positive in the urine of a patient who has undergone prolonged starvation? And you have test number 1, test number 2, and the test number 3. Okay, so your options are 1 and 2 only, 2 and 3 only, 2 only, 1 and 3 only. Now, a patient who has undergone prolonged starvation is likely to have the ketone bodies in the urine. So the correct answer is going to be 2 only. Extra sugar will not be there in the urine, extra protein will not be there in the urine until unless there is some nephropathy and until unless there is a component of diabetes. These two will not be there in the urine. This is the unit test for sugar. This is the heat coagulation test for protein. So here the correct answer is number 2. That is only the Rothera's test for the ketone body. Most widely accepted method for HbA1c estimation is, your options are the ion exchange chromatography, the affinity chromatography, the immunoturbidometry, and capillary electrophoresis. Please note all these 4 methods are used, but the most preferred answer is the ion exchange chromatography, which is considered the gold standard and most widely accepted method for the HbA1c estimation. We'll talk about HbA1c estimation in a separate video. Which of the following is true about mitochondrial DNA? Your statements are higher rate of mutation as compared to nuclear genome, double-stranded circular DNA with 16,000 nucleotides, slight change in nucleotide sequence compared to cellular DNA. Mitochondrial DNA encodes all electron transport chain proteins. Your options are 1 and 2, 2 and 3, 1 and 3, 2 and 4. Please note, higher rate of mutation is correct. 5 to 10 times higher rate of mutation in comparison to nuclear genome is seen for the mitochondrial DNA. Double-stranded DNA, which is circular in nature, not linear, and it has 16569 base pairs. So 16th nucleotides is also correct. 1 and 2 is the correct option about the mitochondrial DNA. Which of the following is associated with defect in formation of the collagen? Okay, your options are rickets, beriberi, scurvy, and pellagra. Also, the answer to this question is scurvy because in scurvy there is vitamin C deficiency. Vitamin C is required for the cross-linking of collagen due to hydroxylation, and in that absence, you have weak collagen, giving rise to what is known as the scurvy. Which of the following test is best for quantitative analysis of the RNA samples? For the qualitative analysis of the RNA sample. Okay, I'll bring it here. Of the RNA sample, your options are real-time PCR, reverse transcriptase PCR, piece restriction fragment length pair technique, and reverse transcriptase followed by real-time PCR. For quantitative analysis of RNA samples, you have to carry out reverse transcription followed by the real-time PCR. The correct answer is option B. Moving to the next question. What is the repair mechanism associated with the CRISPR-Cas9? Your options are non-homologous end joining, the mismatch repair, the base excision repair, and the nucleotide excision repair. So what you have to remember, the correct answer here is non-homologous end joining. We have one whole video on the CRISPR-Cas9, which we'll be discussing later on. We will talk about the concept, the basic principle, the utility of the CRISPR-Cas mechanism, and where it has been already applied successfully clinically. I think this is all that we have from the INI-CET November 2021. Please keep following for all the other papers in the series and the subsequent discussion for the topics which have been highlighted in these papers. Thank you very much. Best of luck and stay watching.